So, so friends, I think it's time we get started. Thangaraj sir, may I know if you're here? Please.
Yes,
Yeah. Thank you. Thank you so much. Yeah. So, uh, so welcome to this Sciversation. Apparently, this is going to be our 76th Sciversation. We have had the first Sciversation this year after almost 6 months of break in between. So basically, to those who are new to Sciversation is making up science and conscience together. This particular forum is largely to take inspiration from eminent scientists and ensure that we learn a large amount of baton from their experience. So basically, we request the uh, guests to quickly introduce themselves within their first 10 minutes or on how their particular life has transcended. But before which, please allow me to take this great honor in having K. Thangaraj, an eminent geneticist and a doyen of Genomics India program, and of course, J.C. Bose Fellow. So just to introduce, to have a very little introduction amid his accolades, Thangaraj obtained his BSc from University of Madras and MSc in 1988, following which he has done MPhil and PhD in 1996. And that was when he joined as scientist B when he finished his PhD, and then later on he moved from scientist C to scientist T1, E2, principal scientist, senior principal scientist, and various accolades has gone through before he becoming the director of CDFD. And of course, Thangaraj and Lalji Singh needs no mention. They have brought in a major impetus to lifelong learning in genetics and as well as genomics for India program. Not only in the annals of various peopling of humans, but also in the areas of other population genetics and as well as diseases. The major research area of Thangaraj was on the molecular basis of CVDs, the cardiovascular diseases, largely in lieu of reproductive health, infertility, and sex determination. But not to mention about the mitochondrial disorders, where a large amount of his focal theme has been into that, thus leading to forensic genetics. Uh, Thangasar received a large number of awards and fellowships. I'll just slightly mention about the prestigious UK India Education and Research Initiative that was awarded by the Gordon Brown, and of course British Prime Minister Fellowship in 2007 to 2011, and Raman Research Fellowship, and later on Rashtriya Ratan Award from Government of India in 2006. And the J.C. Bose Fellowship, which he honors since 2019, has really given a large amount of impetus for him, and over the last couple of years, owing to his retirement, is still in CCMB, working for science, and working for the betterment of human genomics program in India. So thank you so much, sir, for your kind time and energy. I'm absolutely grateful for your time. I'll just probably start with a very basic question. So when you joined CCMB, Lalji was there, and of course, you both make up a wonderful scientific camaraderie. So, what was your wonderful scientific environment that looked like way back in late 1990s, sir?
First of all, thank you, Prashant, for inviting me to this particular program. And it's my pleasure to discuss with you and talk about my research activities for the last 3 decades. As we mentioned, right from the beginning, I used to work with technology thing until the device. And when I joined CCMB, because that was a great shift from my PhD environment to research environment at CCMB, where most of you are aware in the universities, particularly the state universities, where you don't have much of facilities. And when I joined Scientist B, at that time I did not even submit my thesis. Of course, I completed my laboratory work and joined. And it was most of you who have seen CCMB, you know that how the research environment in CCMB is. So for me, first time moving out of the state university lab to the national laboratory, particularly CCMB, at that time was one of the modern laboratories in India. So therefore, that was given a lot more opportunity for me. Of course, technology thing has given full freedom to work. So there was nothing one can say that is not available. So everything which required is available. The only thing is we have to spend time in the lab and generate data and publish. That was the life which I had in the early part of my research career.
Wonderful, sir. I mean, that's quite amazing and fascinating. And I joined CCMB in 2002. The entire 2002, I was there as a young MSc project fellow. And when I just knocked into the gate of CCMB, I had a large amount of goosebumps. That was because I was working with several eminent people, and that was where it's in the bigger cloud of east and west wings of largest group. And of course, I was working with Sunil Verma, but I had a great chance in interacting with you on several occasions. And the kind of CCMB seminars was absolutely fascinating. And I still remember the kind of discussions and the kind of inputs that you would probably give across all the departments. So I'll move to the next point. And you have been leading a wonderful Genome India project, and I've been very fortunate to have reviewed the first thesis of Genome India project of Pritati Kahali. That came up amazingly very good, and sequencing about 9,772 vivid Indian genomes with more than 185 million variants. So if I may just ask you a kind of a gist, what amount of genetic variation that we can be probably proud of with respect to Indian genomes?
Yeah, so since you have reviewed the thesis, you understood so how much of information we can draw from this Genome India project. But for those who are not familiar with this program, I want to give some kind of background. So this is a program which was initiated nearly 5 years ago with the help of DBT, Department of Biotechnology. And Professor Vijay Lakshmi, who was in the Center for Brain Research at IAC, she was the one who initiated, and we all joined hands. There were 20 research institutes which were part of that particular program. 4 institutes, including CCMB, CBR, IGFB, and NIBMG, are the sequencing centers, and there are 16 other centers which were part of it. Some of them have involved in sample collection, some involved in methodology for genome analysis, and so on. So in this particular project, because we don't have any large-scale human genome project, we don't have any background information. Therefore, it was important for us to generate our own data. So most of our study, those who are involved in genetic studies, particularly human genetics, know how unique the Indian genome are, because we have the populations which are very old next to Africans, right? And subsequently we have several migration into India and outside India. Uh, there are several populations which have admixed during the last 2 to 4 thousand years ago. Then there were endogamy practiced for the last 2000 years. And that makes India is one of the very complex nation, having more than 4,600 populations. You can imagine such a huge number of diversity. Staying in India for the last 60,000 years, somehow, of course, have migrated and admixed. But at least for the last 2,000 years, every single population marrying within the community, that's why we as endogamy. That makes every single population is very unique, because the mutation which happened in the last 2000 years is going to remain with a particular population. Therefore, that makes every single population as a unique. And when we compare population from anywhere in the world genetically, we are unique because of all these particular features. Therefore, it is important for us to understand the genetic diversity, understand mutations that are involved in various genetic disease. When you take any mutation associated with the disease, that is very unique to not only to Indian population, but some of the mutations are unique to a particular population. Not you may not find that in any other population. Such a unique population which we have, when we compare some of the mutation, we don't even see that is existing in outside India. Similarly, some of the mutation reported elsewhere, when you try to understand whether that mutation also associated with the same disease which was reported outside India, you may not find most often. Okay. So therefore, it is important for us to understand the Indian genetic variation and the variation that are associated with several diseases. That's the way we have evolved this particular project. Now, 10,000 genome has been sequenced from 83 different populations, and that gave more than 0.5 million variations. And we are now trying to understand the impact of those variations, right? At least more than 1 million variations are probably associated with some kind of disease. Uh, so the disease, broadly we can classify into recessive and the dominant disease. If it is a dominant disease, if the mutation exists in the heterozygous condition, it means we have two copies of the chromosome. Totally, we have 46 chromosomes, exist in 23 pairs. Half of it comes from the father, half of it comes from the mother. If one of the copies carry the mutation, still it expresses the disease, we call them as dominant. But the problem in India is most of the mutations are recessive in nature. Therefore, unless otherwise it exists in both the copies of the chromosome, it won't cause the disease. So now we predicted that most of the population in India are expected to have population-specific recessive disease. Therefore, that can cause population-specific recessive mutation that can cause population-specific recessive disease. That's one of the major problem which we are now trying to understand in stock populations.
Wonderful, sir. Absolutely amazingly narrated. I mean, that also brings us a proposition that, of course, for a larger Indian population like India, sequencing 10,000 Indian genomes is not enough. Probably, we might probably need as many genomes as we could. But at a certainty, at some point of time, we may have to say that okay, we can probably employ some machine learning heuristics. Maybe some 80,000 Indian genomes can probably make up some 120 unique 120 million unique variants, and probably these particular variants can be put up on a machine learning heuristics or probably further imputed in the near future, so that we can identify those rare niche specific variants across specific diseased phenotypes as well. So can we probably think of a kind of a proposition like this, sir, in the near future? Yeah.
Yeah, absolutely. Because as you mentioned that in a country where we have 4,635 populations, 83 population much smaller in number, but at least this is a good beginning where we can understand initially, and there's a lot of scope that it can be extended to many other populations, and we might go into 10,000 become 1 million genome, right? In the future, there's a lot of possibilities.
Right.
Since a lot of variations are existing in the human in Indian human populations, a lot of mutations are very unique. But of course, the mutations that are reported elsewhere, one can understand clearly. But the unique mutations are the one which required, as you mentioned, artificial intelligence and so on to predict and try to understand the impact of those mutations. So, what kind of disease it can cause, and how many populations or how many individuals are going to affect in the future, and so on. Definitely, AI is going to play a major role in the future, particularly where we have huge amount of data. It may take several years, decades for human to understand, but using the AI, we can definitely able to understand very effectively in various amount of time.
Right. Wonderful, sir. So moving further, I mean, my couple of other questions are largely again towards Genome India project. I know we I mean, as a bigger country, we when we started this particular project in 2019, 10,000 was probably felt quite good. But was there any reason why Genome India project couldn't be much focused towards the northeast Indian genomes? Because I could see that very few samples were probably obtained from northeast India, including Kolkata. And second thing was, there's also a major limitation. Probably we could have probably done trios or quads within a family as well. That could have probably given us some kind of an impetus, to understand maybe the recessive allelic conditions like you were probably talking about towards the disease manifestation. Because I since I also work on rare diseases, I mean, the major challenge here is the sample acquisition. Many a time we may not really get the parental genotypes. Thus, we might probably beg to an understanding saying that okay, many of these particular mutations could possibly be autosomal recessive, but in principle, since we don't have the parental genotypes, we might end them marking up as a de novo heterozygous.
Yeah, so it's very interesting that you have brought up this. First, let me start with the trials. In fact, out of these 10,000 samples, close to 10,000 samples, in all 83 populations, we have included 3 to 6 trials, depending upon the population size, sample size. So every single population, we have trials. So that's one point which I want to make it clear. Second one, you said a large number of northeast population. So we had a very long discussion before starting the project. So we wanted to have equal distribution in terms of language. 4 language groups. We have Dravidians, Indo-European, Tibeto-Burman, and Austroasiatic. So that's one of the major. The second one, the social structure, so-called upper caste, middle caste, lower caste tribes and primitive tribes. We also wanted to have equal distribution in terms of the area which India covers all the states and all the geographical regions. Finally, we also want to include the large population, small population which are living in the remote area and so on. So after careful consideration, so we have definitely included Northeast, but it's not so small in number. Right? If you look at any of the brochure or going to website of Genome India, you can clearly see that the samples have been distributed all over India.
Right. Wonderful, sir. Thank you. Sir, moving further, I know there is a major distinction between North India and South India. It is believed that South Indians are more consanguineous, and I would say there's large amount of inbreeding associated with South Indians as well as to North Indians because of a typical Kundalini system in North India, which probably in South India there are large number of castes that are conglomerated, or they might probably marry within the same caste and all those kind of things. So I still remember that wonderful read of yours, that marriage within the same caste is also consanguineous. It's also a typical inbreeding. So, I mean, moving further, if you really see, thanks to the latest sequencing technologies, can we at a certainty, can we say that, okay, in sequencing a human genome, okay, one genome, human genome is about 3.2 billion bases, and for a sufficient depth of coverage, we need at least, say, 25x to 30x depth of coverage. So can we come up with a simple genotyping by sequencing, taking the Genome India project leads that we have taken, which is approximating to 25 to 30x, and cherry pick those particular variants that are linked with consanguinity, and come up with a kind of a panel, and these particular things, and of course could be put up in certain diagnostic centers in the future as genotyped panels, so that it would definitely be very cost effective, because in the near future, although existingly genome patri exists, the kind of sensitivity that the genome patri or the genotyping that was done earlier is not really matching to the latest standards of sequencing. So can we come up in a summary? My question was, can we build up a Genome India Patri kind of thing to narrow down the kind of identifying those typical variants for consanguinity?
So I want to take it little elaborate on this aspect. The main objective of the program, the Genome India program, is to catalog all the variations. The second one is somewhat closer to the one which you are saying, to have an imputation panel. Right? So remember that all the 10,000 samples are unrelated,
Yeah.
and they're all self-reported healthy individuals. Therefore, expecting all the mutations which are responsible for causing the disease is very, very limited, unless otherwise they're all recessive mutation. Okay. But using this as a panel for diagnosis of the disease, it may not be the right choice. Definitely using this as a reference and imputation panel is absolutely fine, which we are trying to make that. Right?
Wonderful, yeah.
That you can get the missing data. But if you wanted to go for diagnosis of the disease, definitely you have to either go for exome sequencing, or if this is a known disease, you can straight away go for a particular mutation, a particular gene. If not, the panel, or if not, go for exome sequencing. Ultimately, finally, you can go for the whole genome sequencing.
Yeah, yeah.
For any approach, I think that's the best thing to do that. But straight away using the Genome India panel may not help for diagnosis of disease.
Right, wonderful, sir. Yeah. Sir, I had a good fortune in moving down south to Kerala, and for 4 years when I was at Amrita University leading a couple of projects. When I landed in Kerala, at the back of my mind, I had something that Kerala embraced matrilineal or matriarchal system during some point of time. And not only Kerala, but some regions of Travancore, Kanyakumari, and Nagarkoil, and some of the districts of Tamil Nadu as well. So, which means there is a large amount of matrilineal system where the progeny obtained is transcended from mother to the girl child, and that's how the system is. So, if you really see some aspects of Kerala or the Malabar coast, there is a large amount of trait or behavioral change that we observe. I know the trait physiology might be associated with a large amount of other things as well, not necessarily specific to what is probably written in genetics. But if you really talk about the Simon's Genome Diversity Project, SGDP project, which you were also associated with that. Simon's Genome Diversity Project is largely across different countries, different subpopulation strata. But here we are talking about a small region of India. So can we come up with an Indian Genome Diversity Project at a certainty? Because in the near future, because of different lifestyles, different behavioral patterns that people are embraced with in India, because the sleeping patterns are varying, we are manifested with large amount of diseases. The average Indian screen time is little more than 9 hours for a human being. So we might probably become something pretty this thing. So there might be some new traits that might even be coming up in the near future, which we really don't know. So can such Indian Genome Diversity Project be taken up in the near future? For example, that I was talking about the matrilineal of Kerala, or could be attributed to the Andaman and Nicobaris peopling that you always talk about from your great nature and science papers and all these kind of things. So where does this particular thing be taken up in the near future, sir?
Yes, definitely it's possible, because the 10,000 genome is a beginning, right? In the future, definitely every single population, I'm sure, will be sequenced, right? So in that angle, as you said, not only the genome sequencing, also epigenetics, particularly the methylation and many other aspects, also is going to be very, very crucial, right? As you said, that different populations are living in different environment. In that scenario, in addition to the genetics, so epigenetics is going to play a major role. Right? Somebody is living in low altitude versus high altitude, and their genome will be different. So their epigenome also will be different. Just as one example. Similarly, the food habit is going to play a major role in terms of their health and many other aspects. So therefore, future, there are sequencing techniques where you can also capture all the methylation profile. So I'm sure that because of the cost per genome sequence is coming down significantly, there will be a possibility that every single population will be sequenced. Now, the central agencies like a DBT has funded for this 10th of the genome. I'm sure in the future, every state also will be taking of the genome of their own state. So I'm sure that it's going to happen soon. So in that case, we will have genome of every single population, if not every single individual, which would help us for understanding not only about their health and disease in terms of genetics, also in terms of the epigenetics.
Yeah, wonderful, sir. Yeah, thank you so much. So moving further, I'll probably switch over to the forensic biology that is more closer to you. I mean, you have extensively worked towards the forensic genetics as well, paving way to wonderful results, not only in the areas of law or also criminology and other genetics. And at the same time, you also have a great sense of humor. I still remember that in every presentation you come up with a great food for thought on how either son-in-law or daughter-in-law is the real culprit, for for the for the child not to be born, for those particular couple. And at the same time, there are several other things where we in India we keep talking about how forensic medicine has really taken up over the last several years. But if you really talk about the forensic medicine, sir, I mean, we hardly find maybe 3 or 4 major institutes. And I know you were responsible for CDFD as well, the launch of CDFD, and of course, along with Lalji, you could bring that wonderful this thing. Can we? Are we? I mean, I was just talking to one of the PhD fellows somewhere in the suburbs of Jaunpur in UP. He says that the the results that are to be taken up in bigger states like UP or in Bihar, if there is a crime that is happening, the DNA fingerprinting or the forensics should should take at least will take at least 3 months time until the reports are obtained. I mean, either they have to send the reports to Delhi or they have to send the reports to something like Hyderabad or Bangalore or major cities, but not across every state capital. And the technology is not that, I would say, not that costly. It is definitely cost effective. We don't need major instruments. But where are we heading? I mean, can't we bring up very good state of the art laboratories so that we can take up forensic medicine at the grassroots levels of every states or probably every districts as well, and ensure that we can bridge the gap in taking up the forensic medicine or forensic genetics to greater heights.
Yes, one of the important application of genomics is a forensic medicine. Right, we can use the genetic variations exist in our genome, because if you look at every single individual have millions of differences. Among the differences which are highly polymorphic is the short tandem repeats. If you take any SNPs, right, maximum you can have only 3 different alleles. Instead of A, there can be G most often or something else. But if you look at the short tandem repeats in a given particular piece of DNA, there will be a tandem repeats, and the repeat number vary somewhere from 5 to 20, right? So that gives more opportunity, more polymorphism. Therefore, every single individual can be differentiated by using these STRs. So that's the basic thing which I want to tell the audience. The second one which you asked, because of the limitation in the laboratories and getting immediate report is a lot of delay, right? I also wanted to inform you that every single state has
Forensic.
state forensic laboratories, but still the number of cases are increasing. Therefore, they could not cope up with the limited manpower. But of course, the whole analysis won't take much time. Once you have the DNA, we have demonstrated that within 3-4 hours you can get the results. But because of the input is more, number of cases are more, they could not able to cope up and give the report as soon. And one of the major uh, aspect which the government, particularly DBT, brought is the DNA bill. So where they wanted to have the DNA profile of all the criminals. And of course, we were in the initial discussion, but later on, it has been transferred to Ministry of Home Affairs. I was there in the few meeting with Ministry of Home Affairs officials, and there's an organization, Indian Criminal Database. I don't remember, but we had a discussion, and they wanted us to make SOP for DNA fingerprinting. Similarly, for many other aspects, we are in the discussion, and they wanted to have a uniform protocol for all the laboratory across India, and everything was going on, and we were being asked to make SOP. We were in the progress of making SOP for DNA fingerprinting. But for some reason, that bill did not get through in the parliament. But I don't know what is the process or progress of that at present. But I'm sure that there's a capability, a technical capability in India, but only thing is we don't have enough laboratory, enough manpower at state forensic laboratory. And to cope up that, if we have more laboratories, as you mentioned, at state, at district level, possible, it won't cost much. But still, there's a lot of process, right, at state level to have more such laboratories. If that happens, I'm sure that the report can be given within 1 day, 24 hours. That is possible because of the robustness of the technique. And that's the technique which have been used worldwide. It's not that only in India, only one laboratory. The STR markers which have been used for forensic application in US or UK or any other country, the same markers are being used. There's no difference. There's no difference at marker level. There's no difference at equipment level. Everything is the same. But the thing is, the crime rate is increasing. Then proportionately, we should have more number of laboratories, more manpower to get the report quick.
Wonderful, sir. Thank you. A final set of questions, sir. I usually ask the guests on are there any ways that you have probably disagreed in an agreeable way with any of your colleagues during your lifetime? This is largely for the younger generation to take inspiration, because the current generation might probably want to learn certain lessons from people like you on how to put disagreements in an agreeable way. So can you just cite one or two examples across your prolific 45 years of your career? Were there any disagreements that you put it in an agreeable way with any of your colleagues?
In scientific, definitely there will be a discussion. The data is the same, and how we interpret, right? So that is different. Most often, there's no disagreement, and only one time we had a discussion extensively, myself, Dr. Lalji Singh, and our collaborator, Dr. Raymond Reach, just before writing the Nature paper.
Yeah.
But otherwise, of course, we came, they also understood what we are trying to say, and that's how the ANI and ASI has evolved,
Yeah.
right?
Yeah. Yeah.
Even today, that is being referred in most of the research publications. So sometimes with extensive discussion goes for not only a few hours, a few days, right? That the better uh, hypothesis, and if you try to convince that your hypothesis is the best, and of course others will agree and accept it. So, but it's very, very few in numbers. Mostly we all agree to
Disagree, yeah.
and the observation.
Wonderful, sir. I mean, I still remember when reconstructing human population history paper was published, Raish et al. Paper. That was quite an amazing read, and we had several journal clubs, to understand that. But even if we really look almost 20 years down the lane, uh, we are to probably identify what is that unique genetic variation that we in India have from these umpteen Indian genomes that have been sequenced. And I'm sure that there are over 4,400 rare diseases that are specifically known in India, apart from the monogenic diseases or sickle cell anemia or any other hemoglobinopathies that we have wonderful testing. I'm sure that there will be enough room for Genome India project to look into the rare disease genomics as well, where we can probably identify what are those specific biallelic variants, or maybe de novo heterozygous variants, or maybe recessive or dominant as well. So one final question
Because
before.
in that regard, just before you come to the
Yeah.
question, in fact, we have another program, major program funded by DBT called pediatric rare disease. When we looked at some of the mutations which are very rare, particularly recessive mutation, when it exists in a homozygous condition, it causes the disease. When we looked at those mutations in Genome India, to our surprise, because these are all supposed to be normal individuals, right? We did find some of them carrying the mutation in heterozygous condition. In recessive disease, heterozygous individuals are absolutely fine. But when you think about the disease and think about the population, then would you expect that if you leave that the mutation in the population without notice, maybe after a few generations, because of the endogamy, that mutation is going to increase its frequency in the population. Maybe after 5 or 10 generations, you might find a homozygous mutation with the disease. So that is why it's very important for us to take all the measures now when we have the mutation in a heterozygous condition. If you put a lot of effort, not only within the scientific community, even with the help of the government, one can able to screen the population, identify the individuals who are carrying the mutation. You can give appropriate counseling, and if the mutations are given to them to monitor in a community, and they can monitor, in fact, either boy or girl who is carrying the mutation, marry the opposite sex. The boy carry the mutation, and the girl also carry the mutation in heterozygous condition. They are absolutely fine now. But there's a chance of 25% of chance that they contribute the abnormal chromosome, abnormal mutation to the offspring. Therefore, there's a disease. So that can be avoided by appropriate screening, appropriate counseling. So that is what is very, very important. And this project like Genome India and pediatric rare disease is going to play a major role.
Yeah, wonderful, sir. Wonderful, very well said, sir. That was, in fact, my question that was probably, leading to that, and of course, we're all very excited as researchers on how this particular thing can be taken up, and I know, and as a person who respects open access, and as of course through Bioclues, where we have been promulgating open source and open access. We look forward for democratization of Indian genomic data so that we can have more and more researchers make a viable use of it, and of course with a due credit given to Genome India partners, and this will probably open enough doors for exhaustive and collaborative research in the area of genomics. Yeah.
No, no, definitely I want to address for this question also. So you maybe remember that DBT has called for the proposal to make use of this 10,000 human genome. The DBT has received few hundreds of the proposals. DBT constituted a committee to evaluate the proposal. And once the evaluation is done, in fact, they will give 50 lakhs or something to carry on. This is one way of accessing. Other way, right now, all the 9,700 and something or 800 sequence have been deposited in IBDC. That's Indian Biological Data Center, which is part of Center for Biotechnology in Faridabad. One can also access directly by writing to DBT and IBDC. And the DBT formed a committee, and the committee will look at the genuineness of the request which comes from EPI. They have to write a proposal, they have to show whether they have ethical committee clearance to do that proposal, show their funding, and will make use of that data. And they should sign an agreement. They will not pass on this to third party. They will not try to identify the individual. All of those. Once that is done, it will be available for the researchers. Right? Definitely, it's going to be available very soon.
Wonderful, sir. So one final question before we request the audience to ask one or two questions. Sir, can we, I mean, I still remember the HaploDB, the haplotype database, specific to humans as well, in the early human genome project phase, way back in early 2000. So if you really talk about what amount of haplo insufficiency that we in India can be attributed to the genomics. I mean, can we come up and say that, okay, there might be some variants that were predominantly pathogenic at some point of time that might eventually, because of genetic drift, they might have gone benign at a certainty, or vice versa. There might be a case where there might be certain benign variants some years ago that might have transcended from generations and that might have eventually turned out to be pathogenic, and the missing link could be haploinsufficiency or maybe triploinsufficiency. So do you see any point here, sir? Yeah.
No, that vary from case to case, mutation to mutation, and that needs to be analyzed accordingly. But according to Genome India project, we are thoroughly analyzing that data, which would be completed soon and be published soon. There will be some information coming out in the paper, but it may not be that extensive in that, because that all of these, you remember, are healthy individuals. So once you take the subjects with the disease, then all of this can be looked at very effectively.
Please, sir. But thank you so much, sir. I mean, I know the time is running up, but we can have one or two questions from the audience. Barka raised her hand. Barka, you have some question?
Yes, sir.
Yeah, please. Yeah.
Good evening, sir. It was such an enriching session. Thank you so much, sir, for enlightening us. It was so good to see and hear the story that how you started with it. Thank you so much. I just have a minor question. Since you have been working in genomics for such a long time, in today's generation, when we have AI and all the ML learning and deep learning softwares, is there anything negative that you feel that's something that you still miss, that this AI or this genomics are always helping us, but something that you feel is at a disadvantage, or something that we are missing using these technologies, and something that you can just suggest at your end.
Uh, when you use any new technology, try to take whatever the best you get out of that, right? Uh,
Yes.
of course, the interpretation one has to do. That's where the human mind goes, right? Otherwise, when you have a huge amount of data, which you difficult for you analyze manually, so that's a plus point of using the AI or ML, right? You can get the information very quickly. So there you can play with the technology, you can play with the software, right? Whatever the best you can get it. Then up you to whether take it as such or modify or ignore it completely. Right? So that's where the human mind required.
Yes, sir. Sir, thank you so much, sir. Yes.
Yeah, thank you, Barka. Velu has a question. Velu, would you like to switch on your microphone and ask? Yeah. Yeah. Sir, so Velumani Selvaraj, who is a postdoc in IIT Kanpur, he asks if endogamy was followed in the south for about 1,000 years or so, it would probably reduce the occurrence of recessive disease mutations in the population due to purifying selection compared to the population in the north. Is this not the reason, or was it observed in this study?
The first part I could understand. What is the second part? First part is asking endogamy is
Yeah,
being is there any recessive
and
disease?
of course, is it due to because of purifying selection compared to the population in the north? Yeah.
Uh, the first part I can tell you clearly that we have identified a few populations which are having a population specific mutation which is totally absent in Genome India data, also absent in 17 lakh chromosomes across the world, right? It's a population specific, and if you go to population, it's very high frequency. Okay, that's mainly because of the endogamy, and also because it's a recessive disease. There's no purification selection there because mainly because it's a heterozygous condition. Individuals are absolutely fine. If it is a dominant, probably it might happen because the individual, also depending upon the severity of the disease, age of onset, and so on. So generalized statement is very difficult. When it comes to recessive disease, there's no purifying selection. It's going to remain the same unless otherwise it gives any kind of disadvantage to the individual.
Right. Wonderful, sir. Thank you so much for your time. We have, of course, Gyaneshwar here, president of Bioclues. And Taruna Rajgopal, yeah, please unmute your microphone, switch on your camera, and please ask the question. Yeah.
Hello, sir. Sir, I just have a question out of curiosity. If you see in most of the developed countries, they have all this sequencing, they have done extensive sequencing of their population. The ultimate aim of doing this sequencing is to have precision medicine, right now, sir. So still they are not able to achieve it. There are some lacunas. What could be the reason, even after doing a lot of extensive sequencing, having a lot of data, what is the reason which is still hindering to achieve precision medicine, sir?
According to several studies, precision medicines are being used successfully for few diseases, right? Including clotting disease, where the vitamin K epoxidase reductase complex, which has been identified, complex gene, so identified as a target gene for warfarin. Okay, and this study is very interesting, which came in New England Journal of Medicine several years ago. So what they have done was they have taken the patient's record and compared with their genotype after sequencing the vitamin K reductase complex 1 gene. And they found there are several mutations, and finally they narrowed down to 7 haplotypes. So haplotype 1, which is defined by a few mutations, and they compared how much of warfarin those individuals have been standardized, because before this gene was identified, the warfarin level was standardized by starting with slow dose, right? And they have been optimized with 3 milligram of warfarin per day. Then there is a set of people with the different set of mutations defined as haplotype 6. And their record shows that they have been optimized with 6 milligram of warfarin per day, 5.9, close to 6. You can now see that without this genotype and just optimized, when they compare with the genotype, some individual require low dose, some individual require high dose, right? Similarly, I think now has been used in the disease like a cancer, cardiovascular disease, and so on. Why it is not been effectively used is that when we don't find a specific target gene for which metabolize the particular drug, and if you use broadly the cytochrome 450 genes, then it's a problem. We cannot actually define a dose or identify a mutation which are responsible for metabolizing the drug. So once it is identified, because we need to work for a little long time with the patient group and the patients which are being treated with a particular drug. So once it's done, then we can probably able to define, okay, so the mutation A in a particular position required such amount of drug, whereas instead of A, there's a G, person required different set of different dose of particular drug. And this is okay with the dose, but sometimes it leads to adverse effect. That is where the major problem. For example, Vaishyas. Vaishyas have pseudocolon esterase deficiency. That is linked with again the mutation. Not everybody, but majority of them. And because of that, if muscle reluctant drug is given when they go for surgery along with anesthesia, they are unable to metabolize the drug, therefore they remain in anesthetic state, therefore it leads to paralysis, right? That's why after some observation in Andhra Pradesh or Telangana, somebody goes for surgery in the major hospital, they ask whether it belongs to Vaishya or not, because for them they give different set of anesthesia and muscle reluctant drug, not the regular one. Okay. So this
Okay.
is under practice, but it takes more time for every single individual to use it.
Yes, sir. That also means that we should look beyond xenobiotic metabolizing genes, right now, sir?
Yeah.
We are more focused on XMEs. We don't look other genes which have a role. We are more focused on only the xenobiotic metabolizers.
Yes, it's required a lot of research, pharmacogenomics, pharmacokinetics, and many more aspects to understand fully, then have a personalized medicine.
Okay, sir. Thank you, sir.
Yeah, what a wonderful answer to a wonderful question, sir. Thank you so much. In fact, it's very interesting that Vaishyas down south are prone to that kind of deficiency, and whereas if you really go down, go up north, Guptas and Agarwals are more prone to be diabetic, and such a vast difference. So, coming to that particular point, sir, I was funded close to 1 crore project by DST on coagulation factors way back in 2019, and we executed that project successfully, and we could identify deterministic markers for clotting factors in Indian subpopulation. So, and you'll be very happy to know that today we are launching this NGS panel, and hopefully this will be launched during INBIX when you are there in Jabalpur ICMR. Yeah, thank you, sir. So friends, I think you all will agree that we had a fantastic Sciversation. We had a mixture of science, conscience, and beyond science and conscience. And I think, I request everyone to switch on your camera for a terrific photograph with Thangaraj sir, and let's make it very everything possible. Maybe in the meantime, you have very familiar faces here, sir. Gyan is here, Mustaq is here, Sugu is here. There are a few members from outside as well, and a few attending as a classroom this thing, which I'll probably put it in recording. And this has been a kind of a great thing. Friends, you'll be very happy to know that Bioclues is honoring K. Thangaraj as a lifetime achievement award awardee for this particular year, and this is going to be held during Indian Conference on Bioinformatics, November 13th to 15th in ICMR Jabalpur, where VM Katoch, Kiran Katoch, also Soumya Swaminathan, and Keshav Singh, and Krishna Mohan. So all these 6 amazing stalwarts are gracing the occasion. So please don't miss attending the conference. And we are also celebrating 20 years of Bioclues, and we want all of you to be there. So just before we take the photograph, I want everyone to stand up and give a standing ovation to none other than Thangaraj for his yeoman service. Yeah.
Thank you to all of you.
Yeah, so even when I'm back to Jaipur, people are very jealous of me because I'm a very good photographer. So I want everyone to switch on your cameras. Subbu always competes with me, so this time I'm not going to give him any chance. Yeah, 1 minute. I'm sorry, I don't know why this is not coming up. Oh, I didn't. Yeah, this unpin. Yes, yeah. Yes, please. Almost all cameras on. Gyan was here. Nayas, Akshay, Mustaq, Velumani, please switch on your cameras, please. Yeah. Yes. The photograph is brighter when we all smile. Yeah. Say cheese. 3, 2, and 1. Yeah. Thank you so much. Thank you so much, everyone. Thank you so much, Thangaraj sir, for your.
Thank you, Prashant.
Thank you so much.
Wonderful discussion.
Thank you, sir.
Thank you. Bye. Bye to everybody.
Yeah. Thank you, sir. Thank you.
Thank you, sir.